It's my pleasure today to introduce today's speaker, Björn Brems. He came over from Berlin today. Björn started his career in Würzburg with Martin Heisenberg, where he was working on Drosophila and already on operant conditioning, which has been a red thread through his whole research career. He then went on to make a postdoc in Houston in Jack Burns' group on a different preparation in aplesia, but still in operant conditioning. And then he came back 2 years ago, now to Berlin with Randolph Menzel, where he's still, or again, working on Drosophila and operant conditioning. And today he's going to tell us a story about learning by doing in the fruit fly, Drosophila. And I'm looking forward to find out what that's about. Björn.
All right, thank you very much. Good afternoon. I've been planning to come and visit for the longest time, so I was really happy and grateful for Thomas when he invited me here and provided me this opportunity to have a look around the campus and visit the diverse groups and people that are working here. And so far, the few hours I've been here, I've thoroughly enjoyed my stay. So thanks a lot. Before I go into the background on learning by doing and those things, I thought I'll start with something that's very general and that most people that are interested in neuroscience are probably going to be well aware of. And that is that our brain and brains in general are energetically extremely costly organs. So for example, the brain makes up only 2% of a person's weight, but it consumes, even at rest, 20% of the body's energy. And for newborns, it's even higher, it's even up to 75%. So that means that the brain consumes energy at about 10 times the rate of the rest of the body per gram of tissue. Actually, this energy consumption has been so large that it's thought that brain size was actually limited by, or mostly limited by, the energy supply that the organisms were able to supply the brain with. Now, so one obvious question is, if it's so expensive and we have to make it larger and larger, where does all that energy go? What's the brain doing with all this energy? And very recently, Marcus Reichel published a paper in Science on resting activity in the human brain, and later also in monkeys. And what you can see here is patterns of correlated activity in the human brain at rest. People call these structures the default network, and Marcus Reichel in this article said something that I thought was very interesting, namely that the energy burden associated with the environment may be as little as 0.5 or 1% of the total energy budget, meaning that the spontaneous activity consumes about 99% of the brain's energy. And whenever then the people in the fMRI are asked to perform a task, there's not the energy demand of the brain doesn't rise more than about 0.5% or 1%. So computing tasks and handling and processing stimuli from the environment apparently only takes about 0.5 or 1% of the brain's total energy budget. So of course it's a very obvious question: what could be so important about this spontaneous activity? And just about a month or two ago, the Reichel group again was able to correlate these spontaneous activity patterns with spontaneous behavioral variability, so that the people were asked to do a button press task, and the force with which they pressed the button varied according to the variability in the brain's spontaneous patterns. So then, of course, the question then is, what could be so important about this sort of spontaneity, about this active behavior that you find in the brain? And then this now leads me to an idea that people have had for a very long time for what activity and active behavior and spontaneity could be important for. And this was about 100 to 120 years ago. In his famous textbook, Principles of Psychology from 1890, William James writes that a curious peculiarity of our memory is that things are impressed better by active than by passive repetition. And we all know that from introspection, that if once we've done things ourselves, we know them much better than if we just perceive them. This idea was popularized a couple of years later by the founder of the Scout movement, Robert Baden-Powell, in his famous book Scouting for Boys. And he said, and that is probably not in the book, but the quote is very enlightening in how he was pushing his idea, and that's he coined the term of learning by doing. In that our educationalists over here are now recognizing that our principle of encouraging active doing on the part of the individual instead of passive reception of ideas by the mass is practical as an education as opposed to theoretical instruction. Now, today, people realize that today's psychologists, they call this this general effect underlying learning by doing the generation effect. The term was coined by Slameka and Graf's landmark paper from 1978, where they showed that if you generate the word psychoneuroendocrinology from a fragment just like this one, you'll remember it better than if you just read the word without actually having to do anything about it. And that's why this effect is called the generation effect, because you have to actively generate the word rather than just passively read it. Later, several years later, almost 30 years later, Cornell and Terrace found the same effect in monkeys. So when monkeys can figure out themselves which button to press and also which button not to press, which image not to press, so are allowed to make mistakes, they learn the task much better and remember it much longer than if they're just shown which buttons they're supposed to press and which they're not supposed to press. This is reminiscent of an effect that in my graduate studies I found in flies. So in flies you can actually teach a couple of things. One of them is, for example, to distinguish an upright from an inverted T as visual patterns. So you can actually teach them, just as the monkeys, they do learn it, it just takes them longer. So you can teach the flies to distinguish between this upright and inverted T, so those performance indices are a measure for how well the flies have learned. And this is, so you can see they do distinguish between the two patterns by simply presenting one pattern and then punishing them with a heat beam, for example, and then presenting the next pattern and not punishing them. So the animals can learn that. However, if they're allowed to explore themselves to find out what is punished and what not, they learn this in about half the time and to about twice the level. So now, in order to study this, one would try to start to dissect and think about what are the components of this system, and then interfere with these components biologically or experimentally, or in another way, experimentally, and then find out where this effect may come from. So if you look at this simple learning, what you have is you have the stimulus, and you have the heat as a reinforcer that instructs the animal as to one pattern being good and another pattern being bad. In this case, you also have the stimulus and the reinforcer. You know, one is good, one is bad, depending on how I set up the heat. But I also have, on top of that, I have the behavior with which the flies control those stimuli. So what you can say is that you have that the stimulus is one component, and stimulus and behavior are two components. You might, if you say that this is active and this is a passive component, or this is a passive procedure, would be the passive procedure, and this would be what we would call the active procedure. However, this active procedure basically just as this one, because here also the word is, the reading is also involved. Whereas the active is not categorically different, it just adds something to the same task. So the active, what we would call an active form of learning, consists of both active and passive components. Now, how can we set up an experiment to do this? To maybe introduce you a little bit deeper into this, because this is very important for the remainder of the talk, into this concept of components in a composite task. you can see that most situations that animals face, be it flies or be it any other animals or humans, is that the animals produce some sort of behavior. The behavior has an effect on the perceived environment. So in the case of the flies, it would be the patterns and the heat. And usually there are certain relationships in the environment. And those are very often called classical relationships, or it's a fact learning procedure that the animals learn that the upright T, for example, is punished and the inverted T isn't, or whatever other visual cues we can use. You will see that flies can learn other things too. So that would be a passive component, or you would call it also a classical component because it's very reminiscent of a task that you all know when Pavlov's dog is learning this relationship between two environmental stimuli. And then in this scheme of things, you'd say that this would be an operant relationship, in that the behavior controls whether or not the heat is being switched on or off or not, or whether or not the animal is being punished or rewarded or something. This would obviously be the active form of learning, and the iconic paradigm for that, of course, is the Skinner box and the rat pressing the lever. And if you've been able to follow me so far, if I haven't lost you yet, then you'll see that the same problem exists here that exists in almost all preparations, in that you can perceive this also as a situation which has operant and classical or fact and skill components, in that the pressed lever signals food just as the bell signals food for the dog. And the technical problem that the people working on rats and mice are having is that you can't take the lever away. So you can effectively isolate the behavior and take the behavior out by simply restraining the animal. That's exactly what Pavlov did. And have it just passively receive those two stimuli. Whereas in this setup, if you take the lever away, there's nothing for the animal to do. So if you want to separate the effects of active and passive components, of fact and skill learning components on learning, you're in trouble if you use this sort of standard learning paradigm. And I'll try to convince you that there's a good way to do this in flies, and that we can find stuff out in flies that you can't, or is a lot more, at least a lot more difficult to do with vertebrates such as rats and mice. So again, you could call this, because it's composed of two components, an operant skill learning component and a classical fact learning component, you could call this composite conditioning or composite learning. I'll try to stick, or I do stick to the color scheme here, that anything that has to do with the behavior, with the skill, will be green. Anything to do with the environment, the facts that the animals learn, will be blue. And then anything that comprises both of these components will be red. So, how did we do this? We use stationary flying flies. So they have a drop of glue between the head and the thorax, and then in this drop of glue is fixed, which is hard to see here, is a little hook, and then a clamp to hold the hook and to tether the fly. You see that the fly is doing all kinds of things. It's moving its abdomen, its legs, even its antennae. And if you put the clamp into something called a torque meter, you can measure a bunch of things depending on what you put into this torque meter. But in this case, you measure the tendency of the fly, its attempts to turn left or right. So if you look at what kind of data you can record, what you see is that the fly is turning left and right. So this is sped up, obviously. This is 30 minutes, a 30-minute experiment. And this is yaw torque. So somewhere in the middle would be flying straight ahead. And you see that the fly changes flight direction seemingly randomly from left to right, basically all over the entire 30 minutes. We have recently published a paper on mathematical analyses of the time structure, the temporal structure of this behavior. And what this analysis suggests is that this is not a random alternation between left and right turning, but that this is actually something that's generated spontaneously by the brain, which looks very random, but is actually a complex combination of nonlinearity and noise. So in order to provide the animal with something to learn, we give it an environment, which is this arena. The arena can be lit either in white, or if we use a color filter here, or 2 color filters, we can illuminate it in different colors. Otherwise, other than that, there are no visual features. So in this case, we won't use patterns, we'll use colors to train the animals. And I hope it's not too confusing to you that the colors are green and blue. That has nothing to do with behavior or fact learning. Those are just the patterns, the colors that are convenient to use for the color spectrum that the animal can perceive. And then to provide an incentive for the animals to actually learn something as a reinforcer or as a punisher, we use an infrared laser diode which heats the fly up. So for example, if the fly is attempting to turn right, which is denoted here by, roughly the middle was straight ahead, so if the fly is turning to the right, we switch the heat off and we turn the arena green. If the fly is attempting to turn to the left, and remember, it's not turning, we really only record its attempts to do that. The fly itself is stationary, as you've seen in the video. We switch the heat on and turn the arena to blue. So now what you have is you have this situation again where you have the colors and the yaw torque, in this case, so the torque around its vertical body axis, the behavior of the animal, which are controlling the heat in a classical or fact learning way and in an operant or skill learning way. So now you have the possibility to experimentally separate the 2 components. Now, how do we evaluate the data? We use performance indices. If they're -1, it means that the fly chose to spend the entire time in the punished situation. If it's +1, it spent the entire period in the unpunished situation. And that means, of course, that if it's 0, the fly did not have a specific preference for a certain yaw torque range or color. And those bars are always 2-minute bars, so every period is 2 minutes long. So these flies almost spent 1 minute in the punished situation and 1 minute in the later unpunished situation. Training means that the laser can be switched on if the right situation arises. The fly chooses the punished situation. And yellow means there's no punishment at all, and it's just a choice or a spontaneous choice test for the fly. So you see that once we switch the heat on, the flies avoid the heat very quickly, and also even when the heat is switched off, they still avoid the punished situation. And now, of course, the question is, are they avoiding the punished behavior, or are they avoiding the punished color? To analyze these processes, we'll always look at the first training at the first test period after the last training, which is marked here as performance index 8. That's the one we're going to look at for the remainder of the talk. Now we just plot that again. Again, as I said, in red, because here we have both the color and the behavior of the fly. That's why in red, the composite situation is just this bar plotted again here. It's just the same thing. So in order to find out, actually let me back up a second. So we already know that for the patterns, if we have the patterns alone, the passive pattern learning, then this is working, or the learning takes longer and isn't as effective as if you have both the color, both the cue, the external cue, and the behavior together. So now what you want to see, and this is basically the same here as well, but now what we want to see is if the symmetrical question is also right. So if I take only the behavior without the colors, does that also lead to a decrease in learning, such that I have the behavior alone, and I have, actually I should do it the other way around. So I have the behavior alone, and I have the colors alone, and they should be lower if it were a symmetrical effect than both together in the composite situation. So we take, this is very simple, we take the color filter out, so the animals don't have the color cue anymore, and do the same experiment. What we find is that the animals can actually learn that. This is both significant in this case. The difference is not significant, but I can show using a slightly altered training regime that this is actually a significant difference, even though statistically this here is not significant. What I did is I reduced the training and then the difference becomes significant. So I have the symmetrical situation in that the two components yield a lower learning score than both components together. And what could be more straightforward now than to hypothesize, well, they two add up. They learn something about the colors, they learn something about the behavior, and then both of them yield a higher learning score. That's very straightforward. So the question is there a summation going on? To test this hypothesis, ideally you just want to knock one of the components out and see how does the behavior, how does the learning pan out. And what you do in Drosophila is you use mutants. In this case you use the rutabaga mutant, which is a very well-known mutant. It's basically been a mutant in any learning paradigm that has been thrown at the animals. And what would you expect? So what is not known though is if this behavioral learning is affected in this mutant. So there's two options basically. So if the mutation, the rutabaga mutation, only knocks out the classical component, the color learning component, which we know is the case, if it only knocks that one out, then you expect something sort of like this, because then only the behavioral learning remains. However, if the rutabaga mutants are just stupid, they don't learn anything, then you'd expect 0 for both experiments. However, what we find is something very different. We find, for one, that this is the first time that rutabaga is wild type at learning anything, so they learn very well the behavioral task. Actually, we could show that it learns it better than wild type. However, the more surprising thing is actually that there's no learning score here, because remember, this differs only from this one by taking the color filter out. So if the animals had eyelids, all they would have to do is close their eyes and then they should do fine. So you have some sort of dominant negative effect of the colors over the behavioral learning. And so the first thing that we showed was that it's not the colors themselves. Just flickering colors, why they learn this kind of behavioral task, does not confuse them. So it's not something trivial in that the flies get easily confused by...
Can I ask you quickly, the rutabaga, they are completely blind or they color blind?
No, they're not even blind. They're just learning mutants. They can see the colors just fine. Yes, okay. They can see just fine. So they have no sensory problems whatsoever, not that I know of. And so for example, they fly straight ahead if you give them a visual panorama. So they have no visual acuity is fine. And despite that their visual acuity is absolutely wild type, they don't learn those patterns, for example. So this is not restricted to the colors. This is really, it's a very general thing that basically and olfaction is the same thing. They've been initially identified as olfactory mutants, that they don't learn smells even though they can distinguish the smells. So it's a very specific mutation for learning, and apparently it's a very specific mutation for classical conditioning. So it's not trivial that the colors just confusing them. The colors are just confusing them. So then what you want to find out to be able to understand what is actually going on is you want to find out at what level does this interference, this dominant negative interference, take place. Are the animals here learning the behavior but they can't show it because the colors are there? Or are the colors preventing any learning from occurring at all? So is it a retrieval or is it an acquisition deficit? And you do this fairly easily experimentally by simply having the standard training here with colors and behavior at the same time, and then after training, so in this case we did the whole procedure without the color filters, but now after the color and behavior training we take the color filter out. Then of course if there's the level of acquisition if the interference is at the level of acquisition, there should be nothing left, right? Because they never even learned anything. Whereas if they did learn something here, they just couldn't show it. If we leave the colors there, then we should get a positive learning score. And this is exactly what we find. So there's interference of the colors at the level of retrieval. When the colors are there, they do learn that left turning is punished, for example, but they can't show that if the colors are there. Now the interesting thing is actually, if you look at the wild type flies, the control flies, they show an interference at the level of acquisition. So if you can learn the colors, then this learning prevents you from learning about your behaviors. And that makes a lot of sense because that's all you need. Once you know green is punished, you can, that's all you need to know. You don't need to know that it's left turning or right turning. So what we have here to sum this up is retrieval and retrieval deficit in the mutant flies and an acquisition interference or inhibition for the wild type flies. So this was done by isolating the operant component, right? The skill learning component, simply by taking this classical component, taking the fact learning component away. So to sum this up, we have a composite learning situation with a skill learning component, which you could call it an active component or an operant component. There's clearly the operant. There's a problem with the nomenclature here, obviously, because operant usually means, in the general term, means this sort of lever pressing thing, which is always something that's composed of several components. So I'll try to refrain from using operant or classical. It'll still, because after so many years it's just ingrained. But I'll try for the rest of the talk to refrain from using operant and classical because it's not really a very adequate nomenclature. And what we found out is that we already knew that the fact learning component is rutabaga dependent, and but we found out that the skill learning component here is rutabaga independent. Now, what we also learned is that the fact learning component inhibits the skill learning component, and it's at the level of retrieval in the mutants, but at the level of acquisition in the wild type flies. What that means, though, is that we said that putting both together leads to enhanced learning. So it's really counterintuitive that one of the first interactions that we find is actually an inhibition. That means that leads us, we must postulate some sort of facilitation that works in the other way. Otherwise you would never get an augmentation, you would never get an enhancement of learning. So we know a little bit now about the biological substrate of those two components. What could be the biological substrate of this inhibition and this facilitation? And for that, it's good to keep in mind this idea that all I have to learn is the colors, because then I can use any behavior to choose between the colors and not just the one I've learned it with, because this is where I'm going to go now. And so what this might, actually to back up here, what this might actually do, it means that I learn this very, very fast, faster than if I had only this fact learning component, and it's independent of what I learned it with. It means I can handle my knowledge, my facts, I can handle them independently, I can generalize them. And this generalization is tested by changing the behavior, so allowing the flies to use a different behavior to access the fact knowledge that they have gained. This is done first by training them in this standard, left and right turning, green and blue mode in this composite situation, and this is just showing that the training here in orange works fine, and then the control flies. And then you transfer a proportion of the flies that have been trained like this after the training in a different situation. And this is, you have to imagine this is sort of like a flight simulator now, in that you have 4 identical stripes in the environment of the fly, and now we feed back the yaw torque, the turning movements of the fly, the left and right turning attempts, by turning the arena. So if the fly tries to turn to the right, we turn the arena to the left, so that the fly gets the impression that it actually turned, by simply changing the visual impression. And when the fly is flying, let's say, towards these 2 stripes, the arena is green, and if it's flying towards the other 2 stripes, we'll make the arena blue. And so we switch the colors right between 2 stripes. So this means that here the flies have to turn to keep the arena green, whereas here the fly has to fly straight to keep the arena green. So basically what it has to do is to do the opposite of what it has been doing during training all along. And if we do that, we see that the flies show no preference for any of the 2 colors. This might be discouraging, but what is very usual, a usual procedure in these cases is that you give them a brief reminder training by simply, for a very brief period of time, let's say 60 seconds, punishing them on the previously punished color, simply to tell them, okay, this is still the same situation. This is something that's done that's very common. And what you find then is you find a nice generalization, significant generalization score. So in this case, now what we've done is we've isolated this classical component by replacing the behavior. So now we have the possibility of isolating each of the two components, look at it in isolation, and perform some biological manipulations with the fly and see what happens. And since this is, as I said, since I said this is a generalization task, because the flies generalize the colors from one behavior to the next, people working in Drosophila, they think of a neuropil in the fly brain which has been associated with generalization, and that's the mushroom bodies. So the mushroom body was the first target to manipulate and to find out what would happen to our composite situation and the two components in it. The mushroom bodies are a paired neuropil structure in the fly brain, which are marked here in green, and you can target most of the cells in the mushroom bodies genetically to switch them off. So you use a little genetic tool, genetic trick to block the output from the mushroom bodies. This is basically equivalent to ablating the mushroom bodies or lesion them. And then we subject them to the same three basically tasks. So we first checked for the generalization. Very firstly, of course, we checked for the composite learning, the skill and fact together, that they learn that fine, and this is something that has been shown before. If you block, output, or ablate the mushroom bodies, the flies appear to be fairly normal. The only thing that these flies are impaired in terms of learning that is obvious and simple is olfactory classical conditioning. For most other learning tasks, those flies are fine. Now what about the other thing? So the blue bar, if you remember, was the generalization bar. And so they performed to our expectation that they did not generalize. So this extends to previous findings that the mushroom bodies are somehow involved in certain cases, not all, but certain cases of generalization. Now interesting, if you look at the skill component, which is suppressed in wild-type flies, this is not suppressed in those mushroom body impaired flies. And this is not a genetic background effect, because the genetic controls, so the parental strains that are pooled here in this case, they reproduce the wild-type results in that they learn the composite situation, they generalize the colors to the different behavior, and they use, presumably, so my interpretation is that it's the suppression of the behavior, of the behavioral learning, of the skill learning component that enables this generalization. So what does that remind us of? It reminds us that this is a very flexible, so I suppress behavioral learning to be flexible. And if I don't suppress behavioral learning, I become stereotyped. I keep turning even though the situation changes. So remember here we just took the color filter out. And they just keep turning to the direction that they've been trained to turn. So that is reminiscent of a habit, right? You do something over and over and over again, and then you keep doing it even though it's not adaptive anymore. So what happens to me all the time is, because we have to unlock and lock our doors at work constantly if we just leave the room because it's all open. I come home at night and I want to open the door to my apartment, and I use my work key all the time. It's just simply, this is a habit that I keep doing all the time. So what if the mushroom bodies were some sort of gatekeeper, were suppressing habit formation in order to enable generalization? Then you would expect this sort of thing to happen if you switch them off. Conversely, if you then train and overtrain wild-type animals, they should show exactly that. They should show that they start learning the behavior, and that should prevent the generalization. And this is exactly what we find. We actually find habit formation. If we just train wild-type flies for twice the regular amount, we get a phenocopy of the flies, so we cannot distinguish them anymore from flies without mushroom bodies or with impaired mushroom bodies. So, to sum this up real quick, mushroom bodies appear to mediate the suppression of skill learning, of the skill learning component, in order to generalize, in order to enable generalization. Repetition can overcome this suppression and form habits. So what does that mean in the red, green, and blue scheme of things? It means that we have a skill learning component which facilitates fact learning. The fact learning in a reciprocal interaction inhibits skill learning. Skill learning is rutabaga independent, fact learning is rutabaga dependent. The mushroom bodies seem to mediate this inhibition. And what I didn't show is that we have good reasons to believe that the mushroom bodies are not at all involved in the facilitation of the fact learning component. And now with this scheme, it's very straightforward. What are the next things that we want to do? Obviously, we want to find out which circuits generate this left and right turning behavior. Obviously, it's like I said, we have good reasons to believe that there are dedicated circuits in the brain that are designed to produce spontaneous behavior, even in the absence of any environmental cues, and we want to find out how those work. Then, of course, if you then train the animals, then you want to find out how is that sensory feedback, how is the heat, for example, how is that fed back into the circuit to modify the circuit so that the spontaneous behavior is biased towards one of the two directions. Of course, since we don't know what the molecular cascades are for operant conditioning, and there's only circumstantial evidence as to what those components might be, or actually preliminary circumstantial evidence for other animals, we want to find out what those operant genes might be. Then what we don't know at all is where is this facilitation being processed. We know it's probably not, most likely not in the mushroom bodies. Where else is it? So that's one of the next questions I'd like to ask. And then, of course, how do the mushroom bodies do this inhibition? So once I found out here how the sensory feedback is integrated, so how this skill learning actually works, and nobody knows that. Once I found that out, I'd like to know how can the mushroom bodies inhibit this sort of learning? And finally, why does that inhibition go away if I just keep training the animals? So that's basically the work of some of that I've done in my graduate thesis in Würzburg. Most of it I've done for the last 2 years in Berlin, and that's the roadmap if I ever get a job and a grant to be able to do that. Thank you.
Yeah, I guess we have time for questions. Yeah.
Yes. So the mushroom bodies are required for some learning, like olfactory learning, but
Yes.
not for this paradigm.
Yes.
So do you think the olfactory learning is a sort of odd paradigm then, and that most of it is mushroom body independent, or is your paradigm the sort of
So it's interesting that it's a little bit heretic what I'm going to say now, and there's a big difference in this classical olfactory conditioning between the training situation and the testing situation. And while it's probably unlikely, I wouldn't completely rule out the possibility that the defect that they have in the classical conditioning is actually a generalization defect, because they have to generalize the odors that they smell during training with the odors that they smell during the test. So it may very well be, although that would be it would be overthrow the dominant, so basically Drosophila neuroscience in terms of learning and memory consists to 99% of olfactory learning. So if that would actually turn out to be true, that would get some eyes rolling. The more likely possibility is, I'd say, is that you have fairly direct olfactory input into the mushroom bodies, whereas at least in the fly, as far as I know, most other input is more indirect. So for example, the visual input into the mushroom bodies, while it's, for example, in bees, it's very well known where that goes in, has, to my knowledge, not been found in flies. So this might might enter the mushroom bodies via a less direct route, and that's why mushroom bodies appear to have a very critical role in olfactory learning directly, but only have very subtle effects. And there's a good reason to believe that the common theme behind the rest of the functions of the mushroom body is inhibitory and as a gatekeeping agent, in that certain stimuli and events are being blocked from entering a memory template. Most likely, I would say, is that you have different populations. That what is known is that the mushroom bodies are not a homogeneous population of neurons. There are several subpopulations, and my best bet would be that a subset of those is highly specialized for classical conditioning, or olfactory conditioning actually, only olfactory classical conditioning, whereas the rest performs other functions.
Well, following up on that, I mean, could you imagine to replace your colors by odors, or is that technically infeasible?
It's in principle, it's possible, but then of course the combination wouldn't work because they don't learn the smell. So there's
for the rutabaga ones, yeah, but
no. But if you delete the mushroom bodies, if you ablate the mushroom bodies, they wouldn't learn it. If it were true that the mushroom bodies, so there is a good friend of mine. He's trying to devise the heretic experiment, namely an olfactory learning experiment where there's no difference between training and test, where the situations are as similar as possible. And if the heretic hypothesis is correct, then they should be able to learn smells, to learn odors. If you assume that the mushroom, a subpopulation of mushroom bodies actually is involved in olfactory learning, then of course you'd have to spare this subpopulation in the experiment you're suggesting to ablate the rest of the mushroom body and leave this odor conditioning subpopulation intact. Then you could do that.
But you could at least do, I mean, without ablating anything, you could do the first parts of your experiments
Yes.
again and see whether it's different for a different sensory modality.
Yes,
right?
you could do it. So I've done some of these experiments in my graduate thesis with patterns, a subset of these experiments, and with wild type flies, and they turn out fairly similar. The problem only is that if you use visual colors, it interferes with the choice of the fly for flight direction, because they try to fly towards the patterns. So, and it's still visual, it's not a different modality. But I'd expect most results in principle to turn out the same way, even if I used a different sensory modality. Yes?
A quick question about the memory test. Are you testing here for short-term or long-term memory?
This is immediately after training.
Immediate.
So there's
Okay.
no, those are continuous. I thought that those, the one I showed initially were like 9 2-minute bars. That's a continuous 18-minute experiment.
So can you speculate on any possible differences between the duration of memory for fact and skill? Just a speculation.
Yes, well, for the memory I have no clue. If introspection and vertebrate experiments are anything to go by, then once you have a behavioral memory, it sticks. It's like riding a bike. Once you have a behavioral memory, it's hard to get rid of. And it would make sense to suppress that sort of learning initially, and to only have this sort of learning occurring when you are dead sure is not the wrong kind of thing. So you can just speculate if you knew how this works in humans, and you could knock this inhibition out with a pill or some other manipulation, you could learn the golf swing in just a few goes, because it wouldn't be inhibited anymore. But of course, if you do that for the wrong behavior, if you do this with children for tying your laces and they tie a knot, they'll be stuck with tying knots in their laces for a long time.
Could you just tell me again, it's just a basic thing, why you prefer this term fact learning and skill learning to the more classical operands?
Actually, the classical I don't have any issues with, because that's easy to separate, and it's easy to make to tell people, okay, it's two stimuli that are associated. It's very easy. With the classical, I don't really have an issue with. The problem is that if you say operant, it always the operant or instrumental learning always involves the environment, and I've noticed that I have a really hard time trying to explain to vertebrate people that we're looking at an operant component in isolation. They have a really hard time just understanding the concept of being able to separate a component in something that they've grown up to see as a monolithic thing, instrumental conditioning that's, you know, that's this sort of, you know, lever pressing rat. And I'm not hell-bent of getting any new nomenclature actually through and get it official or anything. At the moment, I'm only in a position to try to suggest that maybe it's worth thinking or rethinking of what operant conditioning, or what we used to know as operant conditioning, what that actually is, and that it's actually, compared to classical conditioning, it's actually a more complex and composite thing that is very hard to look at from a monolithic perspective, and where separating the components makes a lot more sense.
It's just the term skill also has sort of connotations as well.
It does.
That's the trouble.
It does. There's a very good reason I had this one slide in there where I had active on one side, I had skill learning in the middle, and I had operant on the other side, because I don't really have, I mean, I'm open to suggestions. If there's a good way of solving that without confusing a large proportion of the audience, I'll take it right away. If there's a good solution to that nomenclature problem, I won't argue. But the problem is I see benefits with all the possible versions of the nomenclature, and I have a hard. And of course, I'm not the one who single-handedly decides nomenclature.
No.
So that's why I'm having trouble there. So any suggestions on how to solve that, I'm very open. Or is there a problem?
Well,
Maybe there isn't any.
I don't know. It depends, as you said, it depends a lot on how you grew up with terms. For me, operant has not necessarily the connotation that I'm operating a machine or something. And that's what you don't like about it, right? That you're operating onto the environment and you're not just doing a skill.
Right.
I actually, honestly, personally wouldn't have that connotation, but I believe you that people do.
Nick McIntosh wrote, what used to be here
Yes.
a long time ago,
Oh,
points
yeah.
out these
That's right.
concepts conflated in nearly all experiments on vertebrates and traditional
Yes.
essentially made it.
Yeah, and it's very complicated. Yes.
Have you tried other classic learning memory mutants involved in nanocyclase for in your paradigm? See how
Not yet. We're on the way. So the preliminary evidence from other animals I was referring to was actually from Aplysia, where they find it's a type 2 cyclase and not a type 1 cyclase as rutabaga. And there's a second poster that was just now on the SFN meeting, where they show that in mice it's a type 5 cyclase. I don't think the homologies and orthologies are really very well mapped yet for all of the cyclases. It appears to me that at least both of those cyclases are calcium independent and appear to be dopamine dependent.
have you tried things like dunce and other?
Those standard
Other things
ones,
that
no.
manipulate
Yeah,
those
those we haven't. Now the thing is, of course, while it would be good to corroborate that effect with a different learning mutant, that's a good thing. I'm mostly interested now, to find out. I know it's not the classical pathway. So, which other pathway is it? And it appears again to be a cyclase. It appears to involve PKA and PKC if the other animals are anything to go by. So it might involve some of the downstream components of the cyclase pathways. And then of course it's not clear whether you're dealing with separate neurons or with different compartments in the same neurons. But at least at the cyclase level, the pathways seem to differ. And then of course if you have Duns or amnesic or other of those that are in the same pathway but potentially in different locations of the pathway, we'd have to look that up.
Just returning to the nomenclature, it's just a matter of, if you try to put, I think some very interesting experiments here. When you're trying to publish this, you might get people reading it get hung up on this
Yes.
nomenclature and
I
ignore the
already-
interest in the experiments.
I already got.
Yeah.
I already got on the repeat.
You're as concerned about something about how to define operant conditioning, which is
Yeah.
to some extent.
So what I've tried now, so what I've done in the first version of the paper, so actually I have two versions of this paper up, well it's not a paper yet, it's a manuscript. I have two versions up on Nature Proceedings. One of the versions was trying to be all-inclusive, to say, well this is operant and this is classical, you could call it differently. And I sort of tried to say, to keep the nomenclature constant throughout the paper, but say that, well, it's none of the words I use are really all that adequate and have connotations from different experiments that might confuse the reader. And the referees hated it. So basically, I mean, in the end they said, oh yeah, the nice experiments and we don't have anything scientific to add to the experiment, but we hate the way it's written. And so I wrote it in a new way where there's no operant conditioning, no classical, not a single time appears in the manuscript, and I just stuck to fact learning and skill learning because I figured that's the most general thing that people can relate to, even that are not in the field. And now it's a test, it's an experiment, and now we'll see how it turns out. I have no idea. I just submitted it yesterday. So if you want to compare both of those,
Yeah, I
on
would think
Nature
that we
Proceedings.
can take this discussion to the bar as well. If there's not any other questions, I would think it's off to the IDS bar. Thank you for coming.